Welcome back to Chris Biology, to the series of enzymes. So today we are going to study about the factors affecting enzyme activity. So the activity of an enzyme can be influenced by various conditions that alter the tertiary structure of the protein. These include temperature, pH, change in substrate concentration, and something known as inhibitors. Very, very important. Coming to first two important factors, temperature and pH, these generally have a very narrow range, means that they act at a particular temperature and pH, the activity of the enzyme is maximum, means that is what is known as optimum pH and temperature. So below the optimum pH and temperature or above the optimum pH and temperature, the activity of the enzyme decreases, giving you a bell-shaped curve which looks like this. So the enzyme activity is plotted on the y-axis, and on the x-axis is the pH and temperature. So that you can see the peak, right? That peak is what is known as optimum pH and temperature. So it is different for different enzymes. It is not same for all the enzymes. It varies, but that is what is known as optimum. Below that also the activity decreases. Above that also the activity decreases. Therefore, you get a bell-shaped curve. So next important factor is the concentration of the substrate. So with increasing concentration of the substrate, the activity of the enzyme initially increases, right? But till when does it increase? It increases till it attains a maximum velocity, right? After that it becomes a plateau. It will remain there forever, right? It doesn't change after it has attained maximum velocity. This is because enzyme molecules are fewer than the substrate molecule, and after the saturation of these enzyme molecules, there are no more free enzyme molecules to bind with additional substrate. Therefore, after the Vmax is attained, this is very simple, okay? If all the enzyme molecules are attached to substrate, will there be any left out enzyme molecules? No. Therefore, this is what is known as Vmax. So this is how the graph looks. On the x-axis you have the substrate concentration or S, and on the y-axis you have the velocity of the reaction or V. So on increasing the concentration of the substrate, the velocity of the reaction increases. Till which point it increases? Till it attains maximum velocity. After that it will remain same, it won't change, right? So there is something known as Km, or Km is nothing but the concentration of the substrate at which the velocity of the reaction is Vmax by 2. Please remember, this is not the velocity, this is the concentration of the substrate at which the velocity is Vmax by 2, right? Very important point, mostly repeated NEET question, right? Moving on to study about the last factor. The last factor is the enzyme inhibitors. So enzyme inhibitors can be of two types. Inhibitor is nothing but that shuts off the enzyme activity, means it will inhibit, it will stop the enzyme activity. So it can be of two types, competitive and non-competitive inhibition. In competitive inhibition, there is something like, a person is created same like you. So the same way, the competitive inhibitor is very similar to the structure of the substrate, and it goes and binds to the active site, not allowing the substrate to bind to the active site. So there is a competition in binding of the inhibitor and the substrate. In non-competitive inhibitor, you can see the star is the non-competitive inhibitor. Again, this is an illustration. So this non-competitive inhibitor binds to a site which is known as allosteric site. That is not a active site. It will go and bind to a site other than the active site, which is known as allosteric site, but it changes the structure of the active site. Therefore, the substrate cannot go and bind to the enzyme, right? So this will cause again inhibition of the enzyme activity. But competitive inhibition is a reversible kind of inhibition, while non-competitive is irreversible. Why? Because the shape of the active site itself is changed. So there is no way that you can reverse it, right? That is why non-competitive is irreversible and competitive is reversible, right? So the example that is given in the textbook for competitive inhibition is a very important example. The enzyme succinic dehydrogenase is inhibited by a competitive inhibitor known as malonate, and the substrate here is succinate. Example for non-competitive inhibitors are cyanides and heavy metals. So such competitive inhibitors are often used to control bacterial pathogens. So that was about the factors affecting enzyme activity. Thank you. Please come back to continue with the series of enzyme action.